All right, we're on the record.
Very good. Okay, so our tradition of hearing about the Nobel Science Prizes goes back many years. It started from a discussion with Alan Tartikoff from the medical school about how we could make recognition of the Science Nobel Prizes an interdisciplinary event. Over the years, we've also had co-sponsorship from the chemistry and the biology departments. Today, we're going to hear from my physics colleague, Cyrus Taylor, on the Nobel Prize in Physics, from Thomas Gray of the Department of Chemistry on the Chemistry Prize, and from George Dobiak of the Department of Physiology and Biophysics on the prize in Physiology or Medicine. Because our time is limited, we're asking each speaker to keep to a maximum of 15 minutes so as to allow time for questions. And we'll save our questions until all three speakers have given their segments. Feel free to enter any questions of clarification nature, anything that you have, into the chat box, which Ash will be monitoring. So we start with the 2021 Nobel Prize in Physics. Cyrus Taylor is the Albert A. Michelson Professor in Physics, and his current research is on climate science, which is good because part of the Physics Nobel Prize was given for modeling Earth's climate. Cyrus.
Okay. Is that displaying correctly?
Actually, no, it's in presenter mode right now.
Oh, shoot.
But maybe the same cue as before. Yeah, that's great. It's working now.
Okay, great. Thanks, Phil. It's nice to be here to talk about this. The charge that Harsh gave me way back when was to explain the science and significance of the 2021 Nobel Prize in Physics. And ordinarily, this would be reasonably straightforward. Last year, for example, the prize was given for two discoveries. Discovery of black hole formation as a robust prediction of general relativity, and for the discovery of supermassive compact object at the center of our galaxy. Both are profound, but actually reasonably well circumscribed by the Nobel's description. This year, though, they have an overall theme for groundbreaking contributions to our understanding of complex systems, half of which goes to Shukuro Manabe and Klaus Hasselmann for the physical modeling of Earth's climate, quantifying variability and reliably predicting global warming. And incidentally, that's not a single one because they split that in two. Manabe and Hasselmann are not collaborators together. And then the other half of the prize goes to Giorgio Parisi for a truly breathtakingly broad citation for the discovery of the interplay of disorder and fluctuations in physical systems from atomic to planetary scales. So my goal was going to be to try and parameterize your ignorance of what the prize is given for. Hopefully along the way, we'll actually shed a little light and maybe some insight into the actual physics. But in 15 minutes, this is quite a bit of ground to cover. So let me start with Manabe. He was basically given the prize for having developed the basis for the modern models that we use for modeling climate, general circulation models. And he did this work over a series of decades, beginning in the 1960s. The general question of what's responsible for the Earth's temperature, why does it or other planets have the temperatures they do, actually goes back at least to 1827 to a paper of Fourier, who more or less outlined a program for what we now call climate science that arguably is what's still underway. Manabe's contributions were a couple-fold. First of all, people before had modeled the atmosphere as a series of slabs and looked at radiative transfer from slab to slab at different temperatures. He was the first one to really come in and correctly handle the fact that if you heat a gas from below, you're going to have convection. And that necessarily plays a major role in terms of how the atmosphere as a whole will radiate. Beyond that, a major complication with Earth, of course, is the role of water vapor, which is both a powerful greenhouse gas and not well mixed like carbon dioxide does. It can precipitate. And so arguably his biggest step forward was actually figuring out how to correctly treat the role of water vapor in this very complicated dynamical system. His early work was all just because of limits of computational power in the 1960s, was essentially treating the atmosphere as a single column, a one-dimensional system. By the 1970s, computer power had advanced, and he inaugurated the development of steadily more realistic models incorporating all three spatial dimensions as well as time. So this figure actually is the Nobel Committee, or the Nobel people redrew a key figure from his 1967 paper. So this was one where he investigated what happens if you double or halve the carbon dioxide concentration in the atmosphere. And there are two robust predictions that came out of it. One was that the surface temperature would increase, and the second one was that the temperature of the stratosphere would decrease as you increase carbon dioxide concentration. The choppiness here is basically due to the fact that he only had 17 layers in the model. So you see some of the effects of resolution of that. How well did he do? This again is also from the Nobel presentation. The blue line here is what the models would have if given things like volcanic eruptions that have taken place over the last 100 years or so. If there were no impact as a result of increasing the carbon dioxide concentration. The brownish red one is basically what models that are descendants of Manabe's work say, given the same input parameters, and the black is the data. It's remarkably good agreement. It also illustrates the fundamental variability of the intrinsic variability of the Earth's climate system. That point, the intrinsic variability, is actually where Klaus Hasselmann's work took off. So the Nobel Committee awarded him his portion of the prize for having provided crucial insights into the relationship between weather and climate, and for having developed so-called fingerprinting methods to assess human impact on Earth's climate. So again, his work spanned a number of decades, beginning for our purposes in the mid-1970s with sort of careful studies of what how to characterize the fluctuations in various climate observables on various timescales. Having done that, he then turned to the question of, okay, if we're interested in how it's changing as a result of forcings that we're giving to the system by pumping in more greenhouse gases, how do we address the intrinsic problem of signal to noise ratio in terms of things that we're looking at? And then finally, in the early 1990s, putting all these together, how can you combine multiple signals in such ways to extract as much information from multiple noisy measurements of what you're interested in? This is a figure from his 1976 paper, just illustrating one of the variables where he was looking at what the spectrum of fluctuations, in this case, in sea surface temperatures off of in the Indian Ocean. Just to provide some indication of the kind of challenges that we're actually looking at. He has remarkably few figures in his paper. This is from one of the later ones. That's basically, I think in first approximation, it reduces to say if you've got multiple things you're measuring and you've got a signal that you're interested in on all of them, do a principal component analysis of it. It's not doing full justification to it, but give you some feel for the direction that he was heading people. Now, one of the things is, in the 1970s, when he was doing this work, the signal in climate change due to the amount of carbon dioxide we'd put in the atmosphere was in many ways kind of swamped by the intrinsic fluctuations going on of the Earth's climate system. Since that time, we've pumped vastly more greenhouse gases into the atmosphere. And so things that were matters of astute statistical analysis then actually stick out like a sore thumb now. And so I thought it'd be interesting to look at mean summer temperature. This is Cleveland. The brownish is mean summer temperatures, the deviation around the average between 1961 and 1990. And so you have one histogram, and then the blue is what it's looked like since 1991. Basically, something like a Gaussian distribution, the mean has moved over. You can do the same thing for any place else you pick. I picked Moscow as another example. You see the same effect. And about a dozen years ago, James Hansen basically said, okay, you can basically normalize each location, you can combine the data and see that summer temperatures, as the mean shifts, doesn't feel like it's shifting all that much. But the net result is that things that were happening, once in 1,000 years are now becoming, if not every other year kind of phenomena, increasingly common. And so, Hasselmann was prophetic in the kind of work he was doing. Montaby was prophetic. We're moving into a world that's going to be steadily warmer and warmer. Let me turn now to the second half of the prize. Giorgio Parisi was awarded for the discovery of the interplay of disorder and fluctuations in physical systems from atomic scales to planetary scales. I thought the best way of handling this is just to give you a quick tour through some of his work at sort of the two ends and various places in between. So we start with a 1977 paper with Altarelli. What essentially they did was if you measure the distribution, the momentum distribution of quarks and gluons in a proton, that's essentially a given center of mass energy. How does that change? How does it scale as you go to higher energies? And so this has turned out to be of immense practical importance in terms of, for example, being able to project what you would be able to see at the LHC, given what you knew the distributions were at, for example, the Fermilab Tevatron. Another example is work that he did with Mehran Kardar and Yiqing Zhang on how do interfaces grow in sort of a stochastic environment. One that may not be among his all-time most impactful papers, but one that was impactful on me, was a 1979 paper on random fields, random magnetic fields, supersymmetry and negative dimensions. My first 4 physics papers as a graduate student were essentially all exploring implications of this. On the one hand, in quantum field theory, where it resulted in sort of a new way of doing quantum field theory, and in the context of condensed matter systems, where we looked at correlated random fields and interpreted it in terms of a curved superspace. He's worked on turbulence. He's looked at scale-free correlations in starling flocks, where, likes to tell a story of basically they went through something like 100,000 photos of starlings flocks moving along in terms of understanding the dynamics of it and looking at how the phenomena scale. And then finally, at the planetary scale, he wrote a fascinating paper on stochastic resonance and climate change, which is important in terms of understanding sort of the pacing of the glacial cycles over the last few million years. This paper is interesting in that it was rejected by Science magazine, and it was rejected by the Journal of Atmospheric Science, and of course now has been cited several thousand times. Now, while the actual citation is that extremely broad one, the chairman of the Physics Nobel Committee, in introducing Professor Parisi's Nobel lecture, noted that the Nobel Committee singled out his work on glassy materials, where his papers on replica symmetry breaking and his introduction of the Parisi order parameter have been fundamental to further development of the theory of complex systems. And so I'm going to give you a lightning tour of some of that. So again, he worked this out over a series of papers over more than a decade, and then was returned to it, related work and other problems that are related to this throughout his career. but beginning with infinite number of order parameters for spin classes, then looking at the magnetic properties of these, then looking, coming up with a more intuitive interpretation of what the order parameter is. So what's going on? So those of us in physics are used to dealing with simple models where we have simple spin systems where there's some strength of interaction between, for example, neighboring fields. And then as a function of various parameters, we look at sort of what the collective behavior of the systems are. In these kinds of systems, you have spin systems, that part stays the same, but where the strength of the interactions between any two spins is a random variable, and there's a variety of fluctuations on this. What that means is that, for example, in the kind of things that we have frequently in graduate student courses, you end up looking with at a free energy that may have a single minimum as a function of some parameter, or maybe there's symmetry breaking and you've got something simple like that. These systems, the free energy in a particular sample may look like something in the lower slide. And indeed, from one sample to the next, the statistical properties of the interactions will be the same, but the actual structure of what the free energy looks like as a function of whatever the configuration is will be different. So what does that imply? Well, so the first thing is if you have an energy like that, if you've got a system and imagine cooling it down, then it'll end up in some minimum. On the other hand, and then you can imagine measuring the magnetic susceptibility by turning on a small external magnetic field and seeing how the properties of the system behaves. On the other hand, you can imagine first applying the magnetic field and then cooling the system down, and you'll end up in a different minimum. And so these systems are characterized by actually two different susceptibilities. And the upper one is from one of Piezo's early theoretical papers on this, showing as a function of temperature along the x-axis and magnetization along the y-axis. The lower one being the susceptibility where you cool it down first and then turn on the magnetic field. And the upper horizontal line, horizontal in the idealized model there, is where you turn on the magnetic field first, then cool it down, and then ask what happens with small fluctuations. And then the lower curve is actually the realization of this in an experimental system.
Why is it called the glass? I get the spin, but not the glass.
So basically, there's very long relaxation times associated with it, like actual glasses. I think is probably the best explanation. So bringing it to an end, hopefully I haven't gone over too much. This year's prize is awarded for, and this again quote in the Nobel Committee, for fundamental work on two important problems which only a direct assault on the complex could lead to progress. The first, again, Nobel Committee's language, the complexities of the coupled system of earth, sea, ice, clouds, vegetation, and air, which I've not done justice to here, which determine the Earth's climate, is arguably the most important practical problem now facing science and humanity. And then Piezo's work, particularly on spin glasses, that understanding these, basically the kind of analysis behind this applies to so many other systems in so many different realms that this is probably an example, going back to our conversation before it started, that if there aren't multiple Nobel Prize that comes out of this over the succeeding years and decades, I'll actually be pretty surprised. And so with that, I should bring my part to the end.
Thank you very much, Cyrus. That was very interesting. So now we'll move straight on to the chemistry prize, where we hear from Thomas Gray, Professor of Chemistry. Professor Gray has an extensive research group that focuses on making gold, that noblest of metals, associate with the more proletarian organic compounds to form complexes. And some of these are useful in catalysis. And so he's eminently qualified to tell us about the Chemistry Nobel Prize, which was awarded for work in organocatalysis. Thomas.
Thank you for your introduction, Phil. If gold were a noble metal, we wouldn't have very much to do in my laboratory. So I tell my students they should make gold into an Ig Nobel medal. And so let me tell you a couple things about the Nobel Prize in Chemistry for 2021. It was awarded for the development of asymmetric organocatalysis. Now, that's a mouthful, but it was awarded to a chemist in Germany and one in the United States, Benjamin List at MPI in Germany, and David McMillan at Princeton University in the United States. And both of them are pioneers in catalysis, specifically in organocatalysis, and that part of organocatalysis that's asymmetric organocatalysis. I'll tell you what all these things mean. So this is Benjamin List. He was born in 1968, and he's been around quite a bit. Undergraduate at the Freie University in Berlin, PhD from the University of Frankfurt, and postdoctoral studies and a Humboldt Fellowship at Scripps. And from there back to Germany. He is now at the Max Planck Institute, which is the premier chemistry research institute in Germany, and he holds a number of other significant appointments elsewhere in the world. And he shared the Nobel Prize, it was a 50-50 split, with David McMillan, now at Princeton University. This is another person who's traveled about quite a lot from 2 sites in California over to Princeton. He began his independent career at the University of California, Berkeley, stayed there only 2 years. And from there, he moved to the California Institute of Technology, was there for a little while before he moved to occupy a named professorship at Princeton University. And he now directs a catalytic center there. It is probably an understatement to say that his research spans a wide range of organic chemistry. He was honored with the prize for organocatalysis, but he is also the leading exponent of what is called photoredox chemistry. But for today, I'll focus on his work in organocatalysis. And so what do all of these words mean? So what is a catalyst? A catalyst is an impurity. It's easy to forget that. A lot of people do forget it. But we chemists, when we run a reaction, make a product, we like to purify our product. And that entails removing the catalyst. But a catalyst it's an additive. It's added to the reaction mixture, makes the reaction go faster. And in principle, it can be removed unchanged after the reaction is over. Not consumed in the reaction and can be discovered, but that does not mean that it's a passive spectator. The catalyst is intimately involved in the sequence of steps that leads from reactant to product. It shepherds the reaction mixture through from reactants to products. And the detailed sequence of steps that a reaction goes, starting at reactants and ending in products, is called the reaction mechanism. Okay, and the reaction mechanism is a question of primary importance in chemistry. We want to understand reaction mechanisms for the same reason that an auto mechanic wants to understand the parts of a car, how they all fit together, how they work together. Okay, how a breakdown in one part affects the other parts of the car, because the whole car does not run satisfactorily or at all if all of the parts are not working and if they're not all working in tandem. Similarly with chemical reactions, so it's very important to understand in as much detail as we can the reaction mechanism, the detailed series of steps. Now, it turns out that it's not possible to prove any particular reaction mechanism. We can only disprove it or improve it. A reaction mechanism is at best a hypothesis, but by doing carefully planned experiments, we can gain considerable insight and we can improve our reactions and we can improve our catalysts. Also, many reactions do not go all the way to completion. They do not go all the way to 100% yield of product, but they're held back by an equilibrium constant. They're held back by the fundamental thermodynamics of the reaction. Okay, there's a forward reaction, but there's always a reverse reaction that competes with it. And to the extent that they compete, the reaction may be held back from going all the way to completion. A catalyst does not affect this. A catalyst does not affect the thermodynamics of a reaction. It just makes the reaction go faster. The word itself, catalyst, comes from a Greek word meaning to loosen or untie. It makes intuitive sense. It loosens up the reaction pathway. It makes it go faster and more efficiently. And the concept of catalysis has not always been with us. It was invented by a chemist named Elizabeth Fulhame around 1747. Elizabeth Fulhame self-identified in her publications as Mrs. Fulhame, so I will continue to call her that. And what is the legacy of Mrs. Fulhame? Well, as of when I was in high school, about 1990, Dieter Seebach, who was one of the most famous organic chemists in the world, wrote that new synthetic methods are new ways of doing reactions are likely to occur in biological chemistry and organometallic chemistry. And Macmillan, one of this year's honorees, quoted that in a review that he wrote in Nature in 2008. This is what we call trolling, okay? Because organocatalysis does not fall neatly into either field, biological or organometallic. But I think that's a little unfair because he said this in 1990 and it was substantially true for many years afterwards. And I think it helps to understand this year's prize if we look at it in the context of recent Nobel Prizes in catalysis. Catalysis is enormously important to chemistry. There have been 5 Nobel Prizes, including this one, in catalysis in the last 20 years. And before 2021, the most recent went to Francis Arnold at Caltech for the directed evolution of enzymes. And Francis Arnold's innovation was to take enzymes and to hijack them. Just as a mechanic might toy with the parts of a car, replace one part with a new and better part, Francis Arnold did something similar with enzymes. By a series of directed, precise, purposeful mutations, she was able to get enzymes to catalyze non-naturally occurring reactions. And this is turning out to have huge importance to organic synthesis. The cool thing about enzymes is they're phenomenally efficient and they are selective. They will pick out one and only one substrate, one molecule from the soup and cause it to react. Everything else in the soup, in the biological fluid, gets ignored. And that's a very desirable thing to have, selectivity in catalysis. The metal ion that's involved in Francis Arnold's work is iron. It's heme iron. Heme is in hemoglobin. And the New York Times, in describing her prize, said that she won for a directed evolution of enzymes. Enzymes are proteins that catalyze chemical reactions. She pioneered it. And that is certainly true. Francis Arnold recently joined the Biden administration. She is co-chair with one other person of the President's Council of Advisors on Science and Technology, essentially since the administration took office. And this is a figure that's excerpted from one of Arnold's work. I mean, this is some of her data. This is the business part of enzymes of the type that Francis Arnold engineers. So this is the active site. This orange atom is iron, and it's sitting in a cyclic molecule called a porphyrin. This is heme iron. Okay, heme is in hemoglobin. Heme iron is the reason your blood is red, and it occurs in hemoglobin. It occurs in many enzymes, okay, and it's usually the catalytic site in the enzymes. And so Francis Arnold, through painstaking and precise mutations, can alter the trajectory of these enzymes to cause them to catalyze new reactions and make new products. And so this is the sort of thing that she does. So this iron 4-oxo compound 1, this is a naturally occurring intermediate in biological catalytic cycles. Notice the iron-oxygen double bond. And so in Francis Arnold's hand, she can make instead iron-carbon double bonds, iron-nitrogen double bonds, and each of these does different reaction chemistry. And the chemistry proceeds selectively and with high efficiency. And so this was recognized with the Nobel Prize in 2017. Before that, there was a Nobel Prize in 2010 that was divided among 3 people, Richard Heck, Aichi Negishi, and Akira Suzuki. And this prize was awarded for work done with the metal palladium, using palladium catalysts in new and clever ways to join separate organic molecules together and to carry out transformations that are otherwise difficult to achieve in the synthesis lab. The Times in its write-up said that this work was done in the 1960s and 1970s. And the big thing was linking disparate molecules, parts of molecules that wouldn't ordinarily belong together. Palladium catalysis can make them come together. The Academy highlighted a particular molecule, discodermalide. They had many thousands of others to choose from. And the important thing for our purposes is their catalyst is palladium, so a rarer and heavier metal than iron in Francis Arnold's hands. Before that, in 2005, there was a 3-way Nobel Prize among Yves Chauvin, Robert Grubbs, and Richard Schrock for olefin metathesis. Okay, and the olefin metathesis reaction is another joining reaction. Okay, it's fundamentally different from the palladium catalyzed cross coupling. For olefin metathesis, you need a carbon-carbon double bond. You take the parts of the double bond and you rearrange them. It is environmentally friendly. One application is plastics, and also used to make drugs, many other materials as well. Sadly, Grubbs passed away towards the end of last year, and the Times in its obituary said of olefin metathesis used to make everything from plastics to pharmaceuticals. It's a type of green chemistry, okay, minimal environmental impact. Schock and Chauvin are still living. And going now all the way back to 2001, there was a prize in catalysis, again, a three-way split, William Knowles, Ryoji Naori, and Barry Sharpless for asymmetric catalysis. I'll tell you what asymmetric catalysis is. What their great advance was, they pioneered the ways to make asymmetry happen. And the catalysts they work with were titanium, ruthenium, and rhodium. So everything except the most recent prize involved metal atoms. So let's look at where these metals are. This is iron in the center of the D block. This is where Francis Arnold is. Okay. Titanium is early on in the D block. This is where Barry Sharpless was. And these other metals, ruthenium, rhodium, platinum, these are, palladium, these are all heavier metals, and they are also rarer metals. They're typically more toxic metals as well. And so iron is the most abundant transition metal in the Earth's crust. Aluminum is the most abundant overall, the most abundant metal overall. Iron is the most abundant of those transition metals in the D block in the middle of the periodic table. Titanium, I don't know if you can see my pointer, but titanium is, titanium is up here. There's quite a bit of it too. And ruthenium, rhodium, and palladium, those are very rare metals, rhodium being one of the rarest. So these catalysts that are based on the rare heavy metals, they're not necessarily environmentally sustainable. There's not much of them in the Earth's crust. They're also toxic. Don't forget, a catalyst is fundamentally an impurity. It has to be removed, especially if you're going to inject your product into the human body. Okay, you want to be sure all your rhodium, your palladium, and your ruthenium, they're gone. And so, but what about asymmetric catalysis? Because a big consideration, the prize that went to List and Macmillan was it was asymmetric catalysis. Okay, any object, including molecules, is chiral if it's non-superimposable on its mirror image. Your hands are chiral. Okay, you can't do a perfect superposition of your right hand on top of your left. Okay, and it's actually easy to make a molecule chiral. Okay, one way that often works is to take a carbon atom and bind 4 different groups to the same carbon atom. Okay, and if there's one such carbon in the molecule, that will make the whole molecule chiral. Okay. It's not superimposable on its mirror image, so the molecule and its non-superimposable mirror image, they're called enantiomers. And the object of asymmetric catalysis is to make one chosen pre-selected enantiomer at the expense of the other. Chirality is the rule and not the exception in chemistry. These are just some chiral molecules of an inexhaustible number to choose from. This one on the left, this is the human hormone progesterone. So where these wedges come out of the paper and the dotted wedges go into the page, okay, these are chiral carbons. These 2 drugs, ritonavir and nirmatrelvir, these are the 2 drugs that together make up the anti-coronavirus cocktail Paxlovid. Okay, so several occurrences of asymmetric carbon in each of these molecules. These are also chiral molecules. And so it's very important to organic chemistry to have precise selective ways of making carbon with one predefined configuration at the expense of all others so that we have the right binding geometry. Because not only is the molecule chiral, but proteins are also chiral. That means enzyme active sites are also chiral as well. Okay, the molecule with the correct handedness will bind at the active site. The molecule with the wrong handedness will not. It will be rejected. So it's of huge interest to medicinal chemistry to get your chirality right. And asymmetric organocatalysis is one of the best ways that we have of doing that. Now with organocatalysis, we saw Francis Arnold with irons, Barry Sharpless with titanium, Negishi Heck with palladium and so forth, Schock with molybdenum. Okay, those involved metals. Some of them were heavy metals and rare metals. With organocatalysis, instead of using metals as catalysts, we're using organic molecules as catalysts. Some of them are naturally occurring proteins as catalysts. This avoids the toxicity of the metal ions. And although you still want to purify your compound, it mitigates the toxicity issues that are involved in having a toxic metal ion present at any step of your production. So this is an excerpt from List's first paper on asymmetric organocatalysis, proline catalysis. Proline is a naturally occurring amino acid. You have proline in you, okay, and that catalyzes this reaction. Okay, this image is a bit busy, I realize that. The important thing about it is the two reactants are achiral. Okay, both are superimposable on their mirror image, but the predominant product is chiral. It's non-superimposable. And by choosing the right proline catalyst, he can get the correct handedness of the molecule, the handedness that he wants. This is a space-filling model of proline taken from a biochemistry book. Okay, it's a building block of proteins. This is an excerpt from Macmillan's earliest report of asymmetric organocatalysis. None of these is proline, but many of them are proline-like. And once again, what we have is two achiral reactants, this diene reactant, this dienophile reactant. Okay, both of them are achiral. Okay, both superimposable on their mirror images, but the final product is a chiral molecule. And he was able to achieve this without needing metal atom catalysts. Okay, here we show three catalysts. The usual state of affairs is you have to screen more than 3 molecules before you find a suitably successful catalyst. It's not uncommon anymore to screen thousands of molecules to find one that works or that works well enough. Okay, so 3 molecules is something of an ideal case. And so I think that gives you a flavor of the type of research that led to the Nobel Prize and some context in light of earlier prizes. Catalysis is still and will continue to be enormously important in chemistry. It'll be a major driver of research and development for many years to come. These are some suggestions for further reading if you want them. There's the Nobel Prize website itself. There was a write-up in Nature in 2021. Francis Arnold's entry in Encyclopedia Britannica is quite illuminating. And Mrs. Fulham has her own page on Wikipedia. So thank you for your attention. And I'll be happy when the time comes to take any questions.
Well, thank you very much, Thomas. Very enlightening. And we are recording this, so those links will be available later. Okay, our last presentation is by George Dubiak. He's professor of physiology and biophysics. His research focuses on signal transduction. And so Professor Dubiak is just the right person to tell us about the 2021 Nobel Prize in Physiology or Medicine, as this was awarded for the discovery of receptors that can sense temperature and touch. George.
Thanks very much, Philip. So it's actually interesting. I'm going to be talking about physiology and medicine, the biology, but it actually touches on the two themes that were covered by Cyrus and Thomas, temperature, but temperature sensing in biological systems, and catalysis, because we were talking about types of proteins, ion channels, that catalyze not end chemical reactions, but rather the movement of molecules from one compartment to another. So you can think of ion channels as vectorial enzymes, if you will, that basically speed up a reaction, but in this case, a movement reaction rather than a chemical reaction. So as Philip indicated, on October 4th of last year, so physiology and medicine always comes first of the Nobel Prizes. The Nobel Committee awarded the prize to David Julius and Artem Patapoutian, as Phil indicated, for their discoveries of receptors for temperature and touch. So Dave Julius, you can see he also kind of, we saw, and as Tom Gray indicated, back and forth of some of the Nobel scientists. But this was a relatively small back and forth. Born in New York, then moved to California for his PhD at UC Berkeley, then back to Columbia for a postdoc, back to the East Coast, and then back to California, bumping between East and West, where he's still now at UC San Francisco as a professor. Artem Patapoutian had a little bit longer sojourns from east to west or west to east. I guess it depends on where you want to start. He was born in Beirut, actually right at the peak of some of the very tragic episodes in Lebanese history. He moved to the United States to California and basically stayed there, both for his PhD at Caltech, postdoc at where Dave Julius now is at UC San Francisco. And he's now a scientist, a staff scientist at the Scripps Institute. He's also a Howard Hughes Medical Institute Institute investigator since 2014. So again, as Phil indicated, this is about how do we perceive, how does biology perceive some of the things that Cyrus and Thomas were talking about: temperature, touch, changes in catalysis. And this is actually from the Nobel, publicity release for this Nobel Prize. And this problem goes back quite a ways. So here you can see Descartes thinking about in the 17th century how we might perceive temperature. You put your hand in a fire, you're going to take it away very rapidly. And Descartes really thought about it, that there had to be some sort of mechanical stimulus. He had like strings going up and down our bodies to the brain, and that there'd be some sort of a physical stimulus that you'd pull on the string, and that would sort of set off a bell in the brain that you touch something warm. And it turns out that actually that sort of mechanical is actually not really bad. So, or not a bad analogy, because we now know that through axons, the long parts of nerves that will go, in this case, from the skin to the spinal cord to the brain, it is really a series of strings, if you will, the axons of nerves. And you do have stimuli, and as the Nobel Prize we're going to be talking about today, being able to sense temperature, being able to sense pressure. This is going to involve a series of catalyzed reactions, not dissimilar to what Thomas was talking about in talking about the chemistry. And the Nobel Prize in 1944, going quite a ways back, actually was able to identify different types of sensory neurons that would be able to respond to these changes in physics, if you will: heat, touch, cold. And if you were to look at, say, the ends of our skin, on our fingers, you can identify different regions that are going to be differentially sensitive to harsh touch, to painful touch, or to light touch. And if you actually were to look at that and stimulate these little red dots, which are kind of like corpuscles or the cell endings of nerves, you would see, if you look at electrical signals that would be generated, if you record from the nerve, you'll see changes in the frequency or the amplitude of these ionic electrical changes that are being carried by the nerves up to the brain. And so the prize to Dave Julius and Artem Patapoutian were for the identification of two types of protein complexes, ion channels. Almost everyone in the audience can think back to high school biology, even the non-biologist. And ion channels are just that. These are protein complexes that carry ions from one side of a membrane, of a biological membrane, to the other. And so Dave Julius's contributions were for the identification of temperature-sensitive ion channels that have been given the name TRP channels, transient receptor potential. Again, biology gets its names all over the place. The original TRP channels were identified in fly Drosophila eyes, involved in signal transduction in terms of light perception. And the V is going to be interesting. The V stands for vanillinoid. So now we're going to get back to chemistry. Organic molecules as being part of sort of the sensing material. And you can see these TRP-family channels are involved in temperature, heat sensation, but also nociception or pain sensation. Artem Patapoutian, on the other hand, was really identified another set of ion channels, again, proteins that will catalyze the movement of ions, but in response to changes in mechanical stimulation. And you can see the various types of biological events and functions that these types of piezo channels, piezo coming from the Greek word for pressure. Okay, so how do they do this, and sort of a little bit of the timeline? So some of the timeline goes, again, way back to the 17th, 18th century, to the identification of some chemistry, small molecules from biologicals, isolated menthol, capsaicin. Why do we see, hot peppers being hot peppers? These were identified because we know that these organic chemicals will give a sensation of temperature. Cold for menthol, heat for for hot peppers, capsaicin. And then towards the end of the 20th century, from the 60s to the 1990s, there was a lot of biology done, neurobiology, being able to show that some of these chemicals would trigger different types of neurons to trigger sensations of heat or cold. And that was a lot of biology that was going on. But then in the late 1990s, Dave Julius asked the question, well, okay, if some of these chemicals, these biological chemicals, could trigger changes in neuronal or nerve function that would mimic or replicate the sensation of heat, what might be the biology that can occur? And in a series of papers from the late 1990s to the early 2000s, was able to identify the genes that encoded these ion channel conducting proteins, and that these ion channel conducting proteins were sensitive indeed to temperature. A little bit later, just really 10 years ago now, 12 years ago, Artem Patapoutian asked the same question, but now could there be ways of having ion channels that are going to be sensitive to mechanical deformation? It's going to try to carry you through some of, I think, again, most of you realize or remember back from high school biology that ions are maintained in asymmetric concentrations between one side of a biological membrane, say outside of a cell, and inside. And you can look at any of these ions, but the major ones, potassium's high inside the cell, low outside of the cell. Opposite for sodium, high outside, low inside. And this is information. And by perturbing the flux of these ions across biological membranes, setting it up and then dissipating it, this is information transfer. And this is the basis of nerve-based action potentials. And so you have many, many different types of ion channels that can conduct different classes of ions, cations versus anions, different types of cations, sodium, potassium, calcium. But importantly, you can see that ion channels, in contrast to other types of ion transporting proteins, like ATP-powered ion pumps or co-transporters that will use the ion gradient from one ion to move another ion in the same direction, opposite direction, they're relatively slow in terms of their catalysis of ion flux. In contrast, ion channels have the fastest catalysis because they're relatively dumb molecules, if you will. They open and they close, and that's their job. And so the biology is understanding what are the stimuli that make them gate or go from their open or closed gated. And you can see some ion channels perceive changes in electrical membrane potential. They're voltage gated. Others, when organic molecules bind to them, they will gate either from the outside or the inside. And then of note for Artem Patapoutian's 50% of the Nobel Prize, some ion channels can sense deformation or changes in mechanical gating. And again, ion channels work together. So you can have a voltage-gated ion channel that lets calcium into a nerve. That will cause the release of a neurotransmitter. The neurotransmitter will bind to a neurotransmitter-gated ion channel to start and continue the process. So how does this come now to Dave Julius's work on temperature-sensitive ion channels? So he started with the chemistry or the organic chemistry. He knew that there were neurons, different types of neurons that could be activated. They would open up, they would gate in the presence of capsaicin. What makes a chili pepper, a habanero, or a jalapeno make the sensation of heat? So he took neurons or cells from neurons and basically did a screen and said, okay, if I put capsaicin on these cells that express these putative ion channels, can I isolate the gene that do it? And through this process of knocking out or looking positively for DNA that makes RNA that makes the protein, he was able to identify the first of these temperature-sensitive channels. And it was given the term TRP, transient receptor potential, V, because the capsaicin chemically is a vanillinoid. And this really then set up that he could then take this gene that encoded the vanillinoid, the TRPV4 channel, and then actually ask, okay, without putting the capsaicin on there, if I just change the temperature, will this ion channel open and close? And indeed, that's what he showed. And in a series of studies, both from himself and from many others, this was actually turned out to be the template or the beginning of a family, the first subunit of a family of temperature-sensitive ion channels, which can sense physiological temperature for us as warm-blooded animals in the physiological range. 37 degrees is where we're all sitting at right now, but also noxious heat, up in the 50s, and noxious cold down in the 15 to 20 degree range. And interestingly, in each of the cases of these different TRP family channels, these differentially sensitive to different ranges of thermal sensitivity, that could also be matched by different types of chemistry that would mimic the effects of those different temperature ranges. And so, the Nobel Prize from 2 years ago, or 3 years ago, I guess now it will be this year, was for cryo-electron microscopy, which has revolutionized the study of the structure of biological molecules, especially complex biological molecules like ion channels, because these are proteins with thousands and thousands of amino acids. The ion channel complexes them are actually several of the subunits come together. And here you see the Cryo-EM structures of a couple of TRP family channels. And you can see there's a lot of biology, there's a lot of structure going on there. But getting back to Descartes' whole idea of mechanical sensation is that when temperature, or when these molecules like capsaicin or isolin, which is a menthol derivative, when they actually occupy sites in the structures of these, that will cause changes, conformational changes, mechanical changes in the channel, which will allow it to go from its open to its closed state. And it turns out that when they did the structures of these ions, that where these chemicals, these naturally occurring chemicals, were bind in the structure of these TRP family channels, there's actually endogenous phospholipids there. And so that's really now led to the idea of what can occur here. So here's a very recent paper from last summer for the TRPV3 channel. And you can see, again, you're looking at the electrical signals when you have changes in temperature. So one thing that can occur is you just increase pulse temperature on the cell, and you can see the channel starts to sensitize. In other words, it becomes more and more likely to open in response to the same magnitude of temperature. But at the same time, if you look at the functions of the individual ion channels, you see as you increase from 25 to 36 to 42 degrees, the individual channels will open faster and stay open for longer. And this is really allowed now to get a good mechanical idea of how conformational changes in the temperature, which are being really triggered by changes in how these proteins can detect changes in local temperature or the binding of molecules that mimic the mechanical changes that are induced by heat, open and close the channel. Now, in a very similar, and there's multiple types of ion channels that would be involved in the overall biological response. Now, getting to Arnim Pataputin, he used a very similar, but in many ways, an opposite approach. He knew that there were certain types of neurons that if you measure their electrical membrane potential, you could do that with stick an electrode on or into the cell, and then you used another small pipette that didn't pierce, but just put a little bit of mechanical strain on the cell, you would get a change in ionic current, which is a movement of ions as an electrical signal. And so he was able to then, by knocking out genes and saying, okay, I'm just going to start knocking out and deleting the genes that could encode any potential protein that would allow this mechanical sensation. And then, since now this is in 2010, where we have the entire genomes of mice and humans, we could say, okay, when I knock that gene out, what's its structure? And he was able to identify a family of two genes that encoded proteins that were called Piezo, again, pressure-sensitive ion channels. And again, it's all about the mechanics that's allowing this sensation to occur. So again, using Cryo-EM, which is now revolutionized, as I said, structural biology, you can see this is a very large protein complex. It's actually a trimeric complex of 3 Piezo-1 or Piezo-2 proteins that come together. And what's really remarkable is that parts of the protein that are sort of on the sides of the membrane, spreading their arms out into the side of the membrane. You can see here, looking at from up, down on the channel versus side of the channel, that as you get mechanical strain, that these will act as sensors of how the strain that's dissipated from the lipid bilayer to the protein will cause an opening and closing of the channel. And so you have these sides that are acting as the sensors, and then the middle of the protein is the ion pore. And this is a really active area. And I just checked my Nature site and my Nature feed today, April 14th, 2022, a paper on further structure function analysis of the Piezo-1 channel, again, combining function and structure all together. And so again, you can see how the Nobel Prize to these investigators has just very broad ranging, applications in many areas of biology. And I'll stop there. Thanks.
Well, thank you very much, George. That was fascinating. I'll hand over to my colleague, Professor Matorf, to moderate the questions. But before I do, I'd just like to make this comment that I'd never appreciated that the two meanings of the word hot, piquant and caloric, that it was actually a very good choice to have the same word used in these two things. And what a discovery that what was, I guess, just another sort of use of language gets this verification in this way. So I'm sure there are questions on the three various topics.
Yes.
I don't see anything in the chat. We do have the possibility of giving a reaction by raising your hand.
Right.
I'll hand over to Harsh.
Sure. So I guess there's two basic mechanisms, like Phil said. Either you can raise your hand using reactions, or you can put it in the chat. And we welcome questions, especially from the students, but others as well.
This is Alan Tartikoff. Maybe I could lead off with a couple of questions. One for Dr. Dubiak, and that is, next time I undergo surgery, is it going to be an entirely different ball game with regard to applying certain drugs that calm down these receptors? I've also got a question for Thomas, and that is, why did nobody else get onto this idea of using asymmetric organics as catalysts prior to the individuals whom you described?
Yeah, well, I'll just address Alan's first question, and I'm showing here. You can't see it, you can't read it, but it was one of the reviews that provided some of the figures that I showed. And the title is "Advances in Trip Channel Drug Discovery from Target Validation to Clinical Studies." So yes, identifying additional molecules or synthesizing new molecules, maybe through the organic catalysis that Thomas talked about. This is a big area because these channels, both piezo and the TRP-family channels, are involved in nociception or pain perception. And there are many different targets. Where do you want to get it from? In terms of cancer pain, or inflammatory pain, all the types of pain that the biologists in the audience, or certainly the neuroscientists, study. So this is a big area for drug discovery.
I'll take a stab at your question about organocatalysis. Most organocatalyses function by the catalyst binding the substrate, the molecule whose reaction it facilitates, through hydrogen bonding. And many, if not most, organocatalysis have multiple sites of hydrogen bonding. They can be both hydrogen bond donors and acceptors. There was the perception for a long time that hydrogen bonds are weak interactions. And if you have only one hydrogen bond linking two molecules, yes, that's going to be a very weak linkage, but they can be reinforced by incorporating multiple hydrogen bonding sites in the same organocatalytic sites. Why it wasn't arrived at earlier, I don't have an entirely satisfactory answer to that question. Enzymes are the ultimate organocatalysts, or the ultimate organocatalysts. They're organic molecules. Many of them don't have metals or don't have tightly bound metals that function as organocatalysts, like the serine proteases, for example. But in terms of small molecule organocatalysts, that was a long time in coming. So there may be many people who are kicking themselves over the same question.
So, yeah, I have a question for Tom Gray.
Mm-hmm.
So in Frances Arnold's approach, usually they would take a bacteria expose the bacteria to a new substrate, then they would select through random mutation and see if one of the bacteria has evolved a solution that was the one that was desired. But the question is, so this is a random approach, but to what extent can one now search through computer simulation new catalysts and versus through trial and error?
Computer simulation is a very important part of catalyst screening and catalyst design. Okay, to what extent it's always successful is a matter of discussion in the chemistry community. And I'm not so familiar with Frances Arnold's work that I can tell you accurately what part of her students' effort is consumed in that way, but I'm sure it's not zero. It's computer simulation, and especially with big data, artificial intelligence, it's an important part of catalysis research, and it's only likely to grow.
Thank you.
More questions?
I think I'll have a question for George in this case, if nobody.
Sure, go ahead.
So George, many times Nobel Prizes are given for people who came out with discovery and completely new solutions and approaches, and that opens up the field. So in the discovery of the mechanical receptors, to what extent are these discoveries were basically predictable, given that this research was started in 2000, roughly 2005, 2010. And by then, biology, biophysics, and everything, all the tools were really already available to do a systematic study and eventually find these mechanoreceptors, meaning to what extent is a discovery really worth a Nobel Prize?
Sure. Yeah, I mean, that's obviously going to be a sort of a value judgment. I think, you know, in terms of mechanotransduction, this has been known for a long time, as you note. And in fact, bacteria have mechanosensitive channels. So this is not something that's even with higher order species, if you will, or organisms. But the basis of mechanotransduction was, is the channel itself truly able to sense a change in its in pressure or physical, or rather, does a mechanical stimulus cause something to get leak out of a cell? In other words, for some chemistry to occur, and then the channel basically is sensing the chemistry or the molecule that's being either released or generated. And so I think the reason Pataputian and really got the Nobel Prize is to show that no, the channel has intrinsic sensitivity to mechanical deformation. That would be my spin on the basis of why it was awarded.
Well, thanks. That was a great presentation. Thank you.
And now we have a question from Phil. Go ahead, Phil.
Right. Thank you very much. Yes, this is for Cyrus. So the issue of climate change is a very important one, and the prize was awarded for some very interesting work, although not by people whose life wasn't given to the topic of climate change, but who are developing very interesting, important theories that form the basis of a lot of things one can do. What would you consider to be the unsolved problems in climate change that might lead to a future Nobel Prize?
Oh gosh, you know, if you look at essentially the work that you moved around there, the work that was recognized this time, No, the central part of Manabe's work was essentially starting off by looking at the world as a whole through the single column models, and then moving out to begin to incorporate the the greater complexity associated with the fact that breadth and depth matter as well. And Hasselmann's work, sort of stems from beginning to parameterize the role of climate fluctuation or of weather fluctuations and trying to sort out the signal from them. The direction that much of it's going to go is these are enormously complicated systems. We know that actually fundamentally they're chaotic systems, right? Some of the very simple examples of chaos are from using very, very truncated versions of the basically the equations of motion that go into all of this. The upshot is I think there's enormous room to understand much better any of a variety of phenomena from where we are at the moment. Even I made a very quick pass differentiating between the troposphere and the stratosphere, but even things like understanding in detail the transport properties in the stratosphere is something that we've got an understanding of it at some level, but I think there's enormous room for additional insights. It's an interesting field because there's, on the one hand, right, we've got a wealth of data, though not nearly as much data for almost anything as one would really like. We've got fabulous simulations, but not with nearly the resolution for many things that you would like. And then there's still enormous role for sort of building simplified models to capture the essence of it. And the interplay between these three, I think, is going to continue to be fairly rich for some time to come. That's kind of a roundabout way of saying it, but I think we're more towards the infancy than the maturity of it.
Thank you.
Okay. Well, I see we have a couple of questions. We have a question in the chat, and we have two more participants who want to ask questions. But maybe in light of the time, here's what I propose. We'll end the formal proceedings here and thank the speakers. And then please stick around, everyone, because we'll carry on for a little while and continue the discussion. So thanks very much for the presentations.